learn something about secondary structure prediction. Okay, so now are you aware what is secondary structure prediction? In secondary structure prediction, what do? We try to assign some secondary structural state to the given amino acid residues that are present in our sequence. Whether the first few amino acid residue will form an alpha helix, whether the subsequent 3 amino acid residue will form a turn, so on and so forth. Okay, that would be the ultimate objective of performing secondary structure prediction. Okay, so now let us talk about what are the two different or two major methods by which secondary structure prediction is done. So these two methods are called as alignment based secondary structure prediction method and the methods that are based on single sequences, or what we call it as single sequence based approaches for secondary structure prediction. Okay, so this alignment based secondary structure prediction, the example of the softwares that are based on alignment based secondary structure prediction are PhD, Cypred, JPred, Predator and PSC. Okay, well, these are all online software that you can use online. All you have to do is that you might have to provide the sequence of your protein, and and this software will tell you ki, okay, what might be the secondary structures that might be present in the given sequence. Okay, another type of method are based on single sequence based approaches. Well, say for example, methods like Chow-Fasman and Gore method are based on single sequence. Okay, so what are alignment based secondary structure prediction method and how do they differ from single sequence based approaches? That is what is our next focal point would be. Okay, so now let us talk about this. I have already told you. Okay, protein secondary structure prediction is what? It is nothing but classification of amino acids in a protein sequence according to their predicted local structure. That is what I meant. Okay, well, say for example, if you have this sequence over here, there must be some local short region that यहाँ से लेकर यहाँ तक it is going to be helix, यहाँ से लेकर यहाँ तक it is going to be turn, यहाँ से लेकर यहाँ तक it is going to be strand, यहाँ से लेकर यहाँ तक it is again going to be a turn, यहाँ से लेकर यहाँ तक it is going to be a strand, and these two strand can form sheet, right? बरोबर है? So assigning or classifying the amino acid residue that are present over here according to their predicted local structure, right? What are these local structure? These are alpha helices, turns, strands. The strands can eventually form sheets, right? So this method or this procedure is called as protein secondary structure prediction, or assigning the secondary structure to the given sequence. Okay, so we have already discussed this thing that these local structures can either be of alpha helix, they can be beta sheet, coiled or turns. Okay, so this is what you do in your secondary structure prediction, right? So now I have already mentioned that the secondary structure prediction method can be divided into alignment based and single sequence based method. Okay, so what do we do in this alignment based secondary structure prediction? Okay, so what we do is that we consider the data from homologous sequence or related sequence to assign the secondary structure to the unknown sequence. Okay, so what we do is that we try to find out the family of the sequence in which your unknown, or let's say for example your protein sequence would fit in. Okay, so what we do over here is that, well, say for example, this is your protein sequence जिसमें आपको secondary structure prediction करना है. Okay, now what you do, you try to figure out a protein family in which there would be different members, and you would try to figure out if my protein sequence fit into this family or not, if it has similar sequence to this family or not. Okay, and what we consider is that the secondary structure of the family members is already known, right? We'll say, for example, in this family, all of this family member, यहाँ से लेकर यहाँ तक, there is a helix. Then यहाँ से लेकर यहाँ तक, there is a turn. Then यहाँ से लेकर यहाँ तक, there is another helix. Okay, and then यहाँ से लेकर यहाँ तक, there is another strand, we'll say, for example. Okay, so now you know that whatever members that are present in this family, and you know what is the secondary structural or local secondary structural states of that are present in this family. Okay, now since your sequence is highly similar to the sequences that are present in family, you can, based on this analysis, okay, you can easily say कि since our query protein also belong to this family, that would mean that यहाँ से लेकर यहाँ तक it is entirely possible that it might have a helix. यहाँ से लेकर यहाँ तक it might have turn. यहाँ से लेकर यहाँ तक it might have another helix. यहाँ से लेकर यहाँ तक it might have another strand. Okay, so what do we do over here in this such alignment based secondary structure prediction? The investigator finds the family of a sequence that are similar to the unknown. Okay, so now whatever the homologous region that is present in the family, that is assumed to have the same secondary structures for your query sequence or unknown sequence. Is it okay? So what we are trying to do over here is that we are trying to make prediction that are based on this consensus sequence as such. So whatever, how do we came to conclusion कि यहाँ पर helix रह सकता है? Because we have analyzed multiple sequences that belong to a particular family, and all of these multiple sequences have consensus helix in this region, right? And our sequence is somehow also our sequence is also present in the same family. Therefore, our sequence might also have the same helix as the other members in the same family. Okay? Do you get this concept?
Yes, sir.
Okay, so that is what it means, that the secondary structures, okay, are based on the assumption from the consensus sequence that are present in the family, okay, and not on and these secondary structure predictions are not based on the physicochemical properties of the amino acid residues that are present over here. Okay, so in contrast to this method, there is another method called as single sequence based approaches, in which we do not consider information from any family. We try to deduce the secondary structure of a given protein based on its sequence itself. Okay, single sequence based approach में क्या होगा? We won't consider the information from the family, but we would consider only the intrinsic properties of amino acid residues, whether they can form certain secondary structure or not. Okay, so these are two broad types of protein structure, or you can say secondary structure prediction method. Okay, is it clear now how alignment based approach differ from single sequence based approach?
Yes, sir.
Okay, one more thing that I would like to mention is that whenever we have family of such sequences, okay, what do? We align all these sequences first, and then we can figure out what is the consensus. Therefore, such a type of approaches are called as alignment based approaches. Okay, consensus find out करने के लिए अपने को उनको align करना पड़ेगा ना, right?
Right.
Therefore, these are called as alignment based secondary structure prediction methods. Okay, so now well, let us go into more details of these alignment based secondary structure predictions. So the modern methods for secondary structure prediction exploit information from multiple sequence alignment or combination of prediction from multiple methods, or they can be relied on both of this. Okay, so since we are trying to collect information from multiple sequences from different families of proteins which might be related to each other, the accuracy that is claimed by such alignment based secondary structure prediction methods is very high. Okay, the accuracy usually fall in the range of 70 to 77% over here. Okay, so what happens is that many of these programs are available to be used over web. Okay, there are web servers. What you usually do is that you upload the FASTA format sequence on these web servers. Okay, what web servers I am talking about? These PhD, Cypred, JPred, Predator, PSA, right? So what we do is that we upload our FASTA format sequence of our protein as an input. They will implement their algorithm. They would execute prediction. Okay, and whatever they have predicted, they will send you by your email. Okay, and it is usually done because such predictive stuff can be time consuming as well as computationally intensive. Therefore, once you submit your protein sequence on given such server, it is possible that there might be already few predictions already running over there, right? So it can take some time for you to run your prediction, right? So your sequence might be queued in that protocol or in that pipeline, and whenever prediction is done, they will notify you on your email that your prediction results are ready, and this is how the secondary structures might be in the given protein sequence. Okay, good. Now let us talk about this single sequence based approaches for secondary structure prediction. Okay, so now these were very simplest of its kind. Okay, the very first structure prediction method that was first conceived was back in 1970s, and they were single sequence based approaches. Okay, so Chow Fosman, where this pair was one of they did one of the pioneering work in determining or predicting the secondary structure of given protein. Okay, so what the initial methods like Chow Fosman did was that they tried to devise certain rules from physicochemical data about amino acid residue to predict the secondary structure. Okay, well, say for example, they tried to figure out, this is just an example. Okay, well, say for example, if there is, say for example, if there is term in a given protein 3D structure, so it is, not logical to have a bulky amino acid residue or an amino acid residue which is having a bulky side chain, right? So over here in turns, usually glycines are preferred. Why? Because they don't have bulky side chain. In fact, there is only hydrogen in its side chain. Therefore, glycines would help to stabilize the turns, right? So in that way, they kind of devised rules based on the physicochemical data about the amino acid residue, and they use that physicochemical properties of the amino acids to predict the secondary structure. So now you can say, if there are a stretch of amino acid residues with very small side chains, then it is entirely possible that there might be turnover there. Okay, do you get this concept?
Yes, sir.
Okay, good. So after this pioneering work, okay, Another algorithm was proposed called as GOR. Okay, it was named after the authors Garnier, Robinson, and Osgupthorpe. Some this guy. Okay, I don't know how to pronounce it. To be very frank. Okay, so this GOR algorithm is was further proposed. Okay, and its successor like GOR 1, GOR 2, GOR 3, GOR 4. Well, what these people used is that they used information about the frequency with which the residues occur in helices, sheets, or coils in a protein of known structure to predict the structures. Okay, these people also used, you know, they, they relied on the frequency with which certain residue, might be present in certain secondary structural state. Okay, so when this GOR algorithm was proposed, say few of the structures were already determined, and what did these guys did was that they tried to build up an algorithm, or they tried to build up a consensus ki, okay, how many time is this RG9 present in an alpha helix, or how many time this RG9 is present in a beta sheet, right? What is the frequency of each amino acid residue to happen to be in alpha helix or beta sheets and coil? They call it as propensities of these amino acid residue to take up these secondary structures. Okay, and they use that data for determining the secondary structure of the unknown sequence as such. Okay, so this happened back in 70s and late 70s as such. Okay, now by the time the generation of 80s came, okay, there was sudden increase in popularity of artificial intelligence method. Okay, and these AI based approaches or artificial intelligence based approaches, well, then they shaped alignment based and subsequent secondary structure prediction methodologies as such. Okay, so the subsequent methods exploited similar information as an earlier single sequence based method, but they have done it to automate knowledge acquisition and applications as such. Okay, more advanced predicting algorithms were implemented, like hidden Markov model was used. Okay, well, let us not go in more details of that now. Okay, so how do you measure the prediction accuracy? Okay, so there must be something by which you can measure if whatever software that I have used, what is the level of its accuracy. Okay, so there are two measures that can be used for deciding the accuracy of a given prediction. Okay, the very first method was called as this Q3 method, and another method, or you can say measure, for predicting the accuracy is called as this SOV matrix or SOV method. Okay, so what happens in this Q3 measure for defining the accuracy of your protein, you would calculate, this Q would be calculated as the number of true positive plus number of true negative divided by total number of residues whose prediction has been done. Okay, so higher is the Q value, higher would be the prediction, or accurate would be the prediction. Okay, on the other hand, this another method called as SOF or segment overlap score method, well, that was proposed by these guys, Rost and his co-worker. So what these people relied on is that what they considered is that if they have considered the concept of sparse prediction and good prediction. Okay, so we'll say, for example, if this is the sequence, okay, whose secondary structure was supposed to be predicted. Okay, this is the sequence, right? So now, if your algorithm predicted intact helices and intact beta sheets, okay, that is shown over here. Okay, then such a kind of prediction were given higher score. Okay, on the other hand, if your prediction method predicted something like this, that first residue is going to form a helix, then another residue you don't know, third residue might form helix, and then subsequent two residues you don't know, then fourth residue is predicted to be helix. So such thing never happen in nature, right? Helix is usually contagious. It might begin with this residue and end up with this residue. Okay, so in such case, if there are such sparse prediction, the score of such prediction would decrease. Okay, अगर ऐसा prediction होता होगा तो अपन क्या करेंगे, इसको score कम देंगे, and lower the score, that means the accuracy of prediction is lower. Okay, so such prediction, such sparse prediction would be penalized. अगर ऐसा prediction आता है तो certain score would be minus from the final score, and therefore eventually the final score would then decrease. Final score अगर decrease हो जाता है तो it is not a good prediction. Well, this would be the good prediction. Why? Because we know. Hello, do anyone want to?
यस सर, सर ये नहीं समझ रहा.
नहीं समझ में आ रहा. Okay, so how do we determine if the secondary structure prediction is a good secondary structure prediction or it is a bad secondary structure prediction? So now we know that the helices are always contagious. Okay, ऐसा हो सकता है क्या कि in your sequence this amino acid residue will take a helical conformation while the next amino acid residue will take beta strand?
No sir.
No, right? So if such continuous selection or if such continuous prediction happens, you would give some higher score to this prediction. इसको कुछ वैल्यू देंगे अपन. Okay, and by the end of the entire prediction has been done for the given sequence, you would add up all these scores. You would give some, so say some score x to this, you would give say some score y to this, some score z to this, some score p to this, some score r to this. Okay, and the final score of this prediction would be the combination of x plus y plus z plus p plus r, we'll say for example. Okay, and it would be some number. Okay, यहाँ तक समझ में आ गया?
Yes sir.
Okay, so now if it happens, okay, तो ये number high कब रहेगा? When your algorithm will predict the continuous secondary structures. Do you agree with this?
Yes sir.
Okay. On the other hand, if your algorithm predicted sparse prediction, right? यहाँ से लेकर यहाँ तक हेलिक्स तो रह सकता है, right? यहाँ से लेकर यहाँ तक हेलिक्स तो रह सकता है, but these amino acid residues are not predicted as a helix. Okay? Is it okay?
Yes sir.
What you will do कि ये x में से you would subtract some penalty. Is it okay?
Yes.
Okay, so eventually the score of this x would decrease, right?
Yes sir. Yes.
Score of x would decrease, that means by the end of this prediction the final prediction score का भी score यहाँ पर कम हो जाएगा.
Yes sir.
And eventually जिसका score ज़्यादा, that would be a better predicted secondary structure. जिसका score कम, that would be bad predicted secondary structure as such. Okay, so this is what it is done over here. तो हाई स्कोर किसे दिया जाएगा? टू दिस नॉन ओवरलैपिंग सेगमेंट्स ऑफ सेकेंडरी स्ट्रक्चर. और जहां पर भी स्पार्स प्रेडिक्शन है उसको क्या किया जाएगा? पेनलाइज किया जाएगा. and you would end up something called as segment overlap score. तो जिसका सेगमेंट ओवरलैप स्कोर ज्यादा होगा उसका प्रेडिक्शन एक्यूरेसी ज्यादा है, जिसका सेगमेंट ओवरलैप स्कोर कम होगा उसका प्रेडिक्शन एक्यूरेसी कम है. नाउ इज इट क्लियर?
Yes, sir.
ओके, ठीक है. So now we have dealt with this. Okay, what are secondary structure prediction method, what are their types, and how do we measure the accuracy of secondary structure prediction. Okay, now let us move on to fold recognition. Fold recognition में क्या objective था अपना? We need to determine what might be the fold for the given sequence as such. Okay, that would be the objective of fold recognition. Okay, so let us talk about this method now, something called as fold recognition. Okay, so fold recognition is also called as threading. Okay, fold recognition को दूसरा नाम क्या कहा जाता है? It is also called as threading method. Okay, so the basic principle of structure analysis by threading is that an unknown amino acid residue, unknown amino acid sequence is fitted into, or you can say it threaded into variety of existing structures. Okay, so what would happen is that, we'll say you have got this sequence. Okay, and you have got these 4 or say 3 folds with you. ओके, बात समझ में आ रही है क्या आपको? इसका आपको फोल्ड रिकॉग्नाइज करना है कि भाई ये जो अपना सीक्वेंस है वो कैसे फोल्ड होगा. ओके, एंड यू हैव अ लाइब्रेरी ऑफ से 3 folds नाउ. ओके, so using this threading or fold recognition approach, what you would do is that you would try to thread this sequence into the structure of the fold that you already have. Okay, this is called as what? This is called as threading or fitting the sequence into say certain structure. Okay, and then you would check. हाँ, yes.
ये जो फोल्ड लाइब्रेरी है ये भी वो मल्टीपल सीक्वेंस अलाइनमेंट से ही आएगी?
नहीं, फोल्ड लाइब्रेरी स्ट्रक्चर का रहता है बेटा.
मतलब कैसे सर?
हाँ, मैं बताऊंगा ना.
मतलब हम, अच्छा ठीक.
ओके, सो कंसेप्ट समझ में आ रहा है कि आपको? यू वुड हैव मल्टीपल स्ट्रक्चर्स अवेलेबल, स्ट्रक्चर 1, स्ट्रक्चर 2, स्ट्रक्चर 3, and there would be no number of such structures available. Now your objective would be what? To check out if my sequence fits into these structures. And how would you check it out? By threading it, right? बात समझ में आ रही है आपको? You would try to fit your sequence into this fold.
Yes.
Okay, and then check. You would give some score. Okay, you would check how fit is my sequence into this structure. Okay, and you would give some.
Sir,
हाँ, बोलो.
it's like कि जितने भी अब तक स्ट्रक्चर्स प्रेजेंट है फोल्ड्स उन सब में हम करके देखेंगे और उनको एक एक स्कोर देंगे एंड द highest score will be the most
appropriate fold, right? Okay, or how do we then measure the fitness? Okay, ठीक है, उसके ये पॉइंट के ऊपर अपन थोड़ा बाद में आएंगे. सो व्हाट वी आर गोइंग टू डू इज दैट वी आर ट्राइंग टू फिट और थ्रेड the unknown sequence into numerous structures. Okay, and then we would evaluate their fitness कि भाई अपना सीक्वेंस इसमें ज़्यादा अच्छी तरीके से फिट हो रहा है या ये स्ट्रक्चर में अच्छी तरीके से फिट हो रहा है या ये स्ट्रक्चर में अच्छी तरीके से फिट हो रहा है. Okay, somehow this fitness would be evaluated, and how this fitness would be evaluated ये मैं आपको थोड़ी देर बाद बताता हूँ. ओके ओके. सो नाउ अभी होता क्या है कि दिस थ्रेडिंग मेथड डोंट यूजुअली बिल्ट अप an entire model from scratch. Okay, well, say for example, let us consider we have got our sequence. Okay, अपना sequence क्या है यहाँ पर? You have got, well, say for example, you have got alanine, then you have got cysteine, then you have got aspartate, and you have got, well, say for example, phenylalanine. Okay, in your sequence, and over there you have got this structure. Now in this structure you want to fit your sequence into this structure. Okay, so when you try to predict a fold, इनका क्या कहना है कि एक all atom refined model कभी तैयार नहीं होता है. Okay, so if this is a protein sequence, इसमें भी बैकबोन रहने वाला है और हर एक अमाइनो एसिड रेसिड्यू का साइड चेन रहने वाला है. राइट? Is it okay?
Yes. Yes.
So the structure in which you are going to fit it, it will contain the same atoms of backbone. बरोबर है? The backbone atoms is not going to change whether it is your query sequence or whether it is the protein जिसका fold आपको मालूम है. Backbone atom change होने वाले है क्या? नहीं. Why? Because both of them are proteins. Okay,
Yes, sir.
so when you try to fit this sequence into this structure, इसमें भी कुछ अमाइनो एसिड रेसिड्यू रहेंगे, रेसिड्यू 1, रेसिड्यू 2, रेसिड्यू 3, रेसिड्यू 4, राइट? इसके भी कुछ साइड चेन रहेंगे यहाँ पर, राइट? So what you do is that you replace the side chain that is present on first residue with the side chain that is present in your sequence. बरोबर है? हो सकता है कि द स्ट्रक्चर दैट यू आर यूजिंग एज अ फोल्ड, यहाँ पर फर्स्ट पोजीशन पर हो सकता है कि यहाँ पर Arginine होगा, बट नाउ व्हेन यू ट्राई टू फिट योर सीक्वेंस इनटू दिस स्ट्रक्चर, इंस्टेड ऑफ क्रिएटिंग एन एंटायर मॉडल, व्हाट यू वुड डू, बैकबोन तो सरीखा ही रहने वाला है सबका. ऑल यू वुड डू इज दैट यू वुड ओनली सिंपली चेंज, यहाँ पर जो पहले Arginine था उसकी जगह पर क्या करेंगे, यू वुड सब्स्टिट्यूट विद द साइड चेन ऑफ alanine. डू यू गेट दिस कंसेप्ट?
Yes.
Sir, can you repeat it once again?
ओके, सो दिस ये स्टेटमेंट क्या कहता है कि ऑल एटम मॉडल्स ऑफ द फोल्ड्स आर नॉट प्रेडिक्टेड. तो इसका जो आपको फोल्ड रिकॉग्निशन करना है, तो इसका पूरा एक एटम का आप मॉडल तैयार नहीं करते हैं व्हेन यू डू दिस थ्रेडिंग मेथड. व्हाट यू डू इज दैट व्हाटएवर स्ट्रक्चर टू विच यू आर थ्रेडिंग योर सीक्वेंस, what you would do is that you would only simply substitute the side chain. Everything would be the same.
Right.
Right? And therefore you say that you have fitted the sequence into the structure. Right? So you would
Yes.
have alanine over here. Now you can have, well, say for example, cysteine over here. Cysteine है मतलब यहाँ पर कौन सा ग्रुप रहेगा? सल्फाइड्रिल ग्रुप रहेगा.
Sulfide.
Right? And once you have threaded or fitted your amino acid residues into this particular fold, okay, then you would check for its fitness. Okay, then you would check if my cysteine residue fit properly over here at this location or not. Okay, so you would consider factors like, well, so for example, to what extent the side chain is exposed to solvent. Okay, so well, say for example, over here, if this much portion is exposed to solvent, okay, and in your fitting procedure you have figured out कि over here you have predicted कि यहाँ पर valine रहेगा. So valine is what? Valine is non-polar. Okay, and non-polar residues are not supposed to be present on the surface, right? This valine should be present somewhere over here so that it would remain buried.
Yes.
right? So such a type of fitting is then penalized. ये अगर ऐसे टाइप का फिटिंग हो रहा होगा तो आप क्या करेंगे, फाइनल स्कोर माइनस करेंगे, राइट? इज इट ओके?
Yes.
Okay, so you would consider the intrinsic properties of protein, like polar or charged amino acid residue should be present on your surface. Okay, so after threading your sequence into the fold, you would check this out, ki how many amino acid residue are there that are polar and they are exposed on the surface. If majority of amino acids are in such a way, then you can say that you have properly predicted the fold, right? And if it happens that non-polar amino acid residues are exposed on surface, that means that is not a proper fold for your given sequence. Okay, is it clear?
Yes, sir.
Okay, so well, say for example, you have figured out that there is an aspartate over here. Okay, it is a charged amino acid residue. Okay, that is present in the core of your protein. If such thing can also happen. So now, is there any counter charge like lysine or arginine that can, that can counter charge the, or that can balance the counter charge as such? Okay, बात समझ में आ रही है आपको? So all these things are considered while scoring, or you can say while evaluating the fitting of your sequence into the structure of another fold as such. तो ये मेथड को क्या कहते हैं अपन? We call it as fold recognition or threading. So this is what we are doing. Okay, what we have over here, we have a sequence and we have a template library or a fold library, and what we will do, we would try to fit our sequence into these multiple set of protein and try to evaluate whether they properly fit or not. Okay, if they properly fit, if the score is high, then that means you have identified a fold. You might end up with, say, some structure. बरोबर है? Everyone
Yes.
agrees with this thing?
Yes.
Okay, so in this way you would end up with a fold or a structure. Okay, that is what the scoring scheme over here is. Okay, so this threaded model is scored highly if hydrophobic residue are found in solvent inaccessible environment and hydrophilic residues are found on the protein surface, right? तो इनको अपन क्या करेंगे, ज्यादा स्कोर देंगे. But these high scores are possible only if buried charge and polar residue are found to have appropriate counter charges or hydrogen bonding partners. बरोबर है? Well, say for example, if you have predicted something fold like this, okay, and you have figured out that most of the hydrophobic amino acid residue are present deep buried inside the protein, that means the score would be high, right? तो ये स्कोर हाई कब रहेगा? अगर if there are polar amino acid residue who has got some balancing charges present right over there, काउंटर चार्जेस भी वहीं पर रहने चाहिए, या फिर दिस पोलर अमाइनो एसिड रेसिड्यू शुड हैव सम हाइड्रोजन बॉन्डिंग पार्टनर्स, then and then the score would again increase. Okay, so in this way threading is most profitably used for fold recognition rather than model building. तो इस तरीके से आप क्या करते हैं, you recognize the fold for given sequence. Is it okay? And this is the list of the software that can be used for fold recognition. Okay, there is a server called as Topits, 123D Plus server, 3D PSSM. A popular server that is normally used is Phyre, Phyre. fire. Fire is generally used for fold recognition. So what is the ultimate objective of fold recognition? The ultimate objective of fold recognition is to obtain a predicted fold. Okay, so what we have now? We have the sequence and we have a fold that we have recognized. Okay, since we have a structure available that can be used as a template now, and you already have sequence, you end up with the same situation over here now, right? Again, you can use this technique like comparative modeling or homology modeling and generate the structure for the given sequence. Okay, is it okay? बात समझ में आ रही है आप लोगों को?
Yes, sir.
Yes, sir.
So this is the overall protocol for you to understand कि what method is to be implemented in what condition. Okay, if you clearly find homolog in the given database, you go for homology modeling. If you don't find homolog in PDB, you proceed for secondary structure prediction, then you perform fold recognition. If you are successful in identifying a fold, you can use that fold as a template and perform comparative modeling and generate the 3D structure of a model. Okay, and if you somehow failed at everything, then we will talk about the ab initio structure prediction in the last lecture. Okay, so today you have learned about secondary structure prediction and fold recognition.